Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
All right, let's dive into something pretty cool today. Liver metabolism.
Okay.
Have you ever felt like your body just decides to burn carbs instead of fat? It's like you're trying to be healthy, but your metabolism just have a mind of its own.
Yeah, it's a question that has really fascinated scientists for a long time.
Yeah.
How does our body decide which fuel to use?
Right.
And then what happens when that system kind of goes off the rails?
Well, a new study, it was published in Cell Metabolism, is actually shedding some light on this.
Okay.
And it's pointing to some unexpected players in the liver.
Interesting.
Yeah.
It is really interesting because we always think of the liver itself as like the main organ for processing fat. But this research is suggesting that there's a different story going on here.
Okay. I'm intrigued. Lay it on me.
Right.
What's this new research all about?
So researchers have discovered that specialized cells in the liver called hepatic stellate cells play a much bigger role in fat burning than we previously thought.
Hepatic stellate cells.
Yeah.
Honestly, never heard of them.
Okay.
What are they and why are they so important?
Think of them as like the support system for the liver's tiny blood vessels. They're kind of like parasites, which are cells that wrap around capillaries throughout the body and in the liver.
Yeah.
Hepatic stellate cells, they act as the parasites.
Got it.
And this study found that they are crucial for the liver to switch from burning carbs to burning fat, especially when you haven't eaten in a while.
Okay, so these hepatic stellate cells are kind of like traffic cops. Directing the flow of energy in the liver.
I like that analogy. That's a great analogy. And this study found that a specific protein called plasmalemma vesicle-associated protein, which is found in high amounts on these cells, is absolutely necessary for this switch to happen.
Plasmalemma vesicle-associated protein.
Yeah.
Wow. That's a mouthful.
Yeah, it is a bit of a tongue twister.
Is there a simpler way to refer to that?
Definitely. Let's just call it PVAP for sure.
PVA, much better, much better.
Right.
So tell me more about this PVAP.
Okay, what does it actually do?
Well, we usually think of PVAP in relation to blood vessel health.
Okay.
But in this study, it seems to be acting as a gatekeeper for fatty acids that are trying to enter the liver.
Interesting.
So the researchers removed PVAP from the hepatic stellate cells in mice.
Okay.
And their livers simply couldn't store or burn fat properly, especially when they were fasting.
That's wild. So without PVAP, the liver is basically starved of energy, even if there's fat available.
In a way, yes.
Oh.
The liver is forced to rely more on carbohydrates for fuel because it can't efficiently utilize fat.
Hold on, there's a twist to this story, right?
Yeah.
I read that the mice without PVAP on their hepatic stellate cells actually had better glucose tolerance, meaning they were better at processing sugar.
Yeah.
That seems counterintuitive.
It is very counterintuitive, and that's actually what makes this study so fascinating.
Right. It really highlights how interconnected our metabolic pathways are. Because their livers couldn't efficiently use fat for energy, they had to rely more on burning carbohydrates.
Right.
The study also showed that insulin signaling in the liver was increased in these mice, which further explains their improved glucose tolerance.
Wow. So these tiny cells in this protein we've never heard of could actually have a big impact on how our bodies use energy.
Absolutely.
This is making me rethink how much we still don't know about the body.
I know, right? It's incredible.
There's so much that we're still learning.
Yeah.
And it really is, it raises so many questions.
Yeah.
For instance, have you ever noticed how some people just seem to handle carbs better than others?
Oh, for sure.
This research suggests that individual differences in how our hepatic stellate cells function might actually be part of the reason.
That's a huge aha moment.
Right.
And it makes you wonder, could we target these cells with medicine to help people with conditions like diabetes or fatty liver disease? That's a great question.
Yeah.
And it's definitely a direction this research is pointing toward. If we could find a way to kind of modulate the activity of hepatic stellate cells, or PVAP, it could potentially open up new avenues for treating these really, really widespread conditions.
It's all very exciting.
Yeah.
But it's important to remember that this research was done in mice.
Yes, definitely.
More research is needed to see if these findings hold true for humans.
Absolutely. We can't jump to conclusions just yet.
Right.
And there's another important thing to consider.
Yeah.
The researchers mainly focused on fasting conditions.
Okay.
We still don't know how PVMP and hepatic stellate cells might behave in different metabolic states, like after eating a large meal or during exercise.
So we've learned that the liver's energy source is far more complex than we initially thought.
Absolutely.
But how did the researchers actually figure all of this out?
Yeah.
It sounds like a pretty complicated study.
It is, yeah.
Can we unpack the methodology a bit?
Sure, they used a really clever combination of techniques to pinpoint the roles of hepatic stellate cells in PVP.
Okay, so what did they do first?
So they started by creating mice that were genetically modified to lack PVAP specifically in their hepatic stellate cells.
So they were able to isolate the effect of PVAP in those particular cells.
Exactly.
That's pretty amazing.
Yeah, it's a really elegant approach.
What did they do next?
So then they compared these modified mice to regular mice,
Okay.
looking at a bunch of different aspects of their metabolism. Like they measured the levels of different fats and sugars in their blood, and they even analyzed the structure of their livers under a microscope.
And it sounds like these modified mice showed some pretty significant differences compared to the regular mice.
They did. The lack of PVP in their hepatic stellate cells caused this whole cascade of effects.
Wow.
From changes in how their genes were expressed
Okay.
to alterations in how their livers process fats. And this ultimately led to those metabolic differences that we talked about earlier.
It's incredible how scientists can piece together such a complex story by combining different research methods.
I know, it's amazing.
It's like solving a puzzle.
Yeah, each piece kind of contributes to a clearer picture of how our bodies work at this cellular level.
And in this case, it's revealed a whole new layer of control over our metabolism that we never knew existed.
It really is a paradigm shift in our understanding.
Yeah.
And it's really exciting to see where this research leads us next.
It's pretty remarkable when you think about it.
Yeah.
These tiny cells and this protein play a crucial role in how our bodies utilize energy.
I know.
And that has profound implications for our overall health.
Absolutely. It really underscores the interconnectedness of all these different systems in our bodies.
This research really makes you appreciate the intricate workings of our bodies.
It does. It's truly awe-inspiring.
Okay, so we've talked about how these hepatic stellate cells are like the traffic cops of the liver, directing energy flow. But if the liver can't properly store or burn fat without PVAP, what happens to all those fatty acids floating around?
That's where things get really interesting.
Ooh, cliffhanger.
Yeah.
I guess we'll have to wait for part 2 to find out.
We'll delve deeper into that next time.
That's good.
Great.
Welcome back to our deep dive into the fascinating world of liver metabolism. Picking up right where we left off, exploring the surprising role of hepatic stellate cells in how our bodies use energy.
And remember, we ended part 1 with a bit of a cliffhanger.
We did.
We talked about how these cells, along with a protein called plasmalemma vesicle-associated protein, or PVAP for short, seem to act as gatekeepers for fatty acids entering the liver.
Right. And when the researchers removed PVAP from these cells in mice, their livers couldn't use fat for energy very efficiently.
Right.
So the big question is, what happened to all those fatty acids?
Yeah, where did they go?
Where did they go?
Well, that's where things get really interesting.
Okay.
The study found that instead of just building up in the bloodstream, those fatty acids were actually redirected to the muscles.
Wait, so the muscles were taking on the role of the liver, at least when it came to processing those fats.
Exactly. The researchers saw an increase in fat accumulation in the muscles of the mice without PVAP compared to regular mice.
So it seems like the muscles were stepping in to compensate for the liver's inability to use those fatty acids.
Precisely. And this finding could have some pretty big implications for how we understand the way our bodies distribute and utilize energy. It suggests a level of flexibility and communication between organs that we maybe didn't fully appreciate before.
It's like our bodies have this really intricate network of checks and balances, ensuring that energy gets where it needs to go, even when one part of the system isn't functioning optimally.
That's a great way to put it, and it opens up so many fascinating questions. For example, could this redirection of fatty acids to the muscles have any long-term effects? Could it be beneficial in some ways, or could it potentially lead to problems down the road?
Yeah, I was just thinking the same thing. On the one hand, it's pretty amazing that our bodies can adapt like this. But on the other hand, you have to wonder if there are consequences to kind of pushing the muscles to handle a job they weren't necessarily designed for.
Exactly. It's a classic example of how scientific discoveries often raise more questions than they answer.
Right.
And that's what makes research so exciting. It's a constant process of exploration and discovery, peeling back the layers of complexity.
Okay, so we've talked about the kind of fascinating implications of this finding.
Yeah.
But I'm also curious about the techniques the researchers use to make these discoveries. Can we dive into the methodology a bit? I remember you mentioned they use some pretty advanced genetic engineering.
Absolutely. One of the key techniques they used is called conditional gene knockout. It's a really powerful tool that allows scientists to very precisely delete specific genes, but only in certain types of cells.
So in this case, they were able to target just the hepatic stellate cells and remove the gene responsible for producing PVP.
That's exactly right. And this precision is crucial because it allows researchers to isolate the effects of a gene in one specific cell type without disrupting its function in other parts of the body.
It's amazing to think that we have the tools to manipulate genes with such precision. It's like having a microscopic scalpel that can target individual cells.
That's a great analogy. And it really highlights the power of modern genetic research. By using techniques like conditional gene knockout, scientists can gain incredible insights into the roles of specific genes and how they contribute to complex processes like metabolism.
So by removing the PVAP gene specifically from the hepatic stellate cells, the researchers were able to see how that impacted the liver's ability to use fat for energy.
Exactly. And they combined this technique with a range of other methods, including gene expression analysis, to really understand the intricate mechanisms at play.
Can you remind us what gene expression analysis is and why it's so important in this context?
Sure, gene expression refers to the process by which our DNA is used as a blueprint to create proteins. By analyzing which genes are active or turned on in different cells, we can learn a lot about what those cells are doing and how they're responding to different conditions.
So in this case, the researchers looked at gene expression in the livers of the mice to see how the absence of PVP was affecting the overall activity of the liver.
Exactly. And what they found was really interesting. The lack of PVAP caused significant changes in the expression of genes involved in fat metabolism, which provided further evidence that these hepatic stellate cells play a key role in regulating how the liver utilizes fat for energy.
So it's not just about whether PVAP is present, but also about how it influences the activity of other genes involved in the whole process of fat metabolism.
Right. It's a complex network of interactions, and this study has really shed light on the role of PVAP as a kind of conductor, orchestrating the activity of multiple genes involved in fat metabolism.
This is also fascinating, but I'm also realizing that we've been focusing primarily on the molecular level, looking at genes and proteins.
Right.
Did the researchers also use any techniques to visualize what was happening in the liver at a larger scale?
They absolutely did. In addition to the genetic and molecular analyses, they also used various imaging techniques to get a clearer picture of what was happening in the livers of these mice.
That makes sense. Seeing is believing, as they say.
Exactly.
So what did they see when they looked at the livers under a microscope?
Well, one of the most striking observations was the difference in fat storage between the modified mice, lacking PVA, and the regular mice. They could literally see the accumulation of fat in the livers of the regular mice, but this accumulation was much reduced in the livers of the mice without PVAP.
So the visual evidence mashed up with the molecular findings, confirming that without PVAP, the liver wasn't storing fat as effectively.
Exactly. And this visual confirmation was really important because it helped to strengthen their conclusions.
It's amazing how scientists can piece together such a complex story by combining these different approaches, from manipulating genes to visualizing cells under a microscope.
It really is a testament to the power of scientific inquiry and the ingenuity of researchers. And this study in particular has given us a whole new appreciation for the intricate workings of the liver and its crucial role in managing our body's energy balance.
It's also highlighted the crucial role of these often overlooked hepatic stellate cells, which seem to be much more than just structural support for the liver's blood vessels.
Absolutely. They're emerging as key players in the regulation of fat metabolism, and this discovery has opened up a whole new area of research with the potential to lead to new treatments for metabolic disorders.
And on that note, I'm eager to explore those potential implications for treatment and delve deeper into the broader significance of this study. We'll tackle all of that in Part 3, so stay tuned for the final installment of our deep dive into the fascinating world of liver metabolism. Welcome back to the final part of our deep dive. We've gone through the intricate workings of the liver, met the unsung heroes called hepatic stellate cells, and learned about this protein called plasmalemma vesicle-associated protein.
Right.
But now I want to know, what does it all mean, in the big picture?
Yeah, this research has some pretty big implications for how we understand metabolism and how we might think about treating metabolic disorders in the future.
So we're talking about new potential treatments for like diabetes and fatty liver disease.
Exactly. So remember how those mice, the ones without plasmalemma vesicle-associated protein on their hepatic stellate cells were actually better at handling sugar?
Yeah.
That suggests that targeting those cells or the protein itself could be a new way to improve blood sugar control in people with diabetes.
That sounds so promising. And what about fatty liver disease?
Yeah.
Could this research help us tackle that?
I think there's definitely potential there.
Okay.
We know that the buildup of fat in the liver is a major driver of fatty liver disease.
Right.
So if we can find ways to help the liver use fat for energy more effectively, it could help prevent or even reverse this condition.
So these hepatic stellate cells and this plasmalemma vesicle-associated protein could really be the key to unlocking new treatments for these disorders.
It's definitely a promising avenue for research.
It's exciting.
It is exciting, but of course more research is needed to make sure this translates to safe and effective treatments for humans.
Yeah, that's a good point. It's easy to get carried away with the possibilities,
Right.
But we have to be patient and let the research run its course. But even beyond the potential for new treatments, this study has really changed how we think about the liver.
Absolutely. It challenges the traditional view that the liver is the only organ involved in fat metabolism. And it shows how important these hepatic stellate cells are.
Right.
We often overlooked them before.
Yeah, who knew these little cells were so important?
I know, right? They're like the unsung heroes of the liver.
This brings up a lot of new research questions,
Yeah.
We don't fully understand how hepatic stellate cells communicate with other cells in the liver, right?
Exactly.
And what about the long-term effects of sending all those fatty acids to the muscles? Is that sustainable?
Yeah, good questions.
Could it have unintended consequences for muscle health?
Yeah, those are some of the really important questions that future research needs to address.
It's amazing how one study can completely change how we see something as fundamental as metabolism. It makes you appreciate the power of scientific research and always being curious.
Absolutely. It shows how important it is to explore the unknown, even if it means challenging what we thought we knew.
This research is a perfect example of that. It took us on this journey from tiny cells to the complex ways that our organs work together and energy. It's all connected.
It's all connected.
So as we wrap up our deep dive into this study, what's the key message you want our listeners to take away?
I think the most important takeaway is that our bodies are incredibly complex.
Yeah.
And interconnected. What seems like a small change in one type of cell can have big effects throughout the entire system.
And those effects can have a huge impact on our overall health. Wow.
Exactly. This research has opened our eyes to a whole new level of control over our metabolism. And as scientists keep exploring, who knows what other amazing discoveries are out there.
It really is an exciting time to be following this research, and we can't wait to see what the future holds.
Yeah, me too. Thanks for joining us on this deep dive.
And to our listeners, thanks for listening. We hope you enjoyed learning about the fascinating world of the liver and metabolism with us.
Stay curious, everyone.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.